Hello everyone, and this is Sanskriti Nagarkar, and today we are going to read the lesson, the chapter 2, Biological Classification from Unit 1, that is the diversity in living organisms. Okay, so in this we are going to learn about kingdom Monera, kingdom Protista, kingdom Fungi, kingdom Plantae, kingdom Animalia, and then about viruses, viroids and lichens. Since the dawn of civilization, there have been many attempts to classify living organisms. It was done instinctively, not using criteria that were scientific, but born out of a need to use organisms for our own use, for food, shelter and clothing. Aristotle was the earliest to attempt a more scientific basis for classification. He used simple morphological characters to classify plants into trees, shrubs and herbs. He also divided animals into two types, those which had red blood and those that did not. In Linnau's time, a two-kingdom system of classification with Plantae and Animalia kingdoms was developed that included all plants and animals respectively. This system did not distinguish between the eukaryotes and prokaryotes, unicellular and multicellular organisms, and photosynthetic green algae and non-photosynthetic fungi organisms. Classification of organisms into plants and animals was easily done and was easy to understand, but a large number of organisms did not fall into either category. Hence, the two kingdoms classification used for a long time was found inadequate. Besides gross morphology, a need was also felt for including other characteristics like cell structure, nature of wall, mode of nutrition, habitat, methods of reproduction, evolutionary relationships, etc. Classification systems for the living organisms have hence undergone several changes over the time. Though plant and animal kingdoms have been a constant under all different systems, the understanding of what groups or organisms be included under these kingdoms have been changing. The number and nature of other kingdoms have also been understood differently by different scientists over the time. So this is table about the characteristics of the five kingdoms. Now which are the five kingdoms? Monera, Protista, Fungi, Plantae, Animalia. I'll repeat again: Monera, Protista, Fungi, Plantae, Animalia. The cell type of Monera is prokaryotic, cell type of Protista is eukaryotic, cell type of Fungi is eukaryotic, cell type of Plantae is eukaryotic, and cell type of Animalia is also eukaryotic. Cell wall of Monera is non-cellulosic, that is, it is made up of polysaccharide plus amino acids. Cell wall of Protista is present in some of the Protista organisms present in Protista kingdom. Cell wall of Fungi is present and it is made up of chitin, and the cell wall of Plantae is present and it is made up of cellulose, and cell wall of Animalia is absent. Nuclear membrane of Monera is absent, nuclear membrane of Protista is present, nuclear membrane of Fungi is present, nuclear membrane of Plantae is present, nuclear membrane of Animalia is present. Body organization of Monera is cellular. Protista body organization is also cellular. Body organization of Fungi is multicellular or of loose tissue. Body organization of Plantae is either of tissue or organ, and body organization of Animalia is of tissue or organ system. Mode of nutrition of Monera is autotrophic. In that autotrophic it is either chemosynthetic or photosynthetic, and the second type is heterotrophic, that it may be saprophytic or parasitic. The mode of nutrition of Protista is autotrophic, in that it is photosynthetic and heterotrophic. Mode of nutrition of Fungi is heterotrophic and saprophytic. In that heterotrophic it is either saprophytic and parasitic. Mode of nutrition of Plantae is autotrophic and it is only photosynthetic, and mode of nutrition of Animalia is heterotrophic, in that it is either holozoic or saprophytic, etc. Okay, so R.H. Whittaker in 1969 proposed a five-kingdom classification. The kingdoms defined by him were named as Monera, Protista, Plantae, Fungi and Animalia. The main criteria for classification used by him include cell structure, body organization, mode of nutrition, reproduction and phylogenetic relationships. Table 2.1 gives a comparative account of different characteristics of the 5 kingdoms. The 3-domain system has also been proposed that divides the kingdom Monera into 2 domains, leaving the remaining eukaryotic or kingdoms in the 3rd domain, and thereby a 6-kingdom classification. You will learn about the system in detail at higher classes. Let us look at this 5-kingdom classification to understand the issues and consideration that influenced the classification system. Earlier classification system included bacteria, blue-green algae, fungi, mosses, ferns, gymnosperms and angiosperms under plants. The character that unified this whole kingdom was that all organisms included had a cell wall in their cells. This placed together groups which widely differed in other characteristics. It brought together the prokaryotic bacteria and the blue-green algae, that is cyanobacteria, with other groups which were eukaryotic. It also grouped together the unicellular organisms and the multicellular ones. Say for example, Chlamydomonas and Spirogyra were placed together under algae. The classification did not differentiate between heterotrophic group, fungi, and the autotrophic green plants, though they also showed a characteristic difference in their walls composition. The fungi had chitin in their cell walls, while the green plants had a cellulosic cell wall. When such characteristics were considered, the fungi were placed in separate kingdom fungi. All prokaryotic organisms were grouped together under kingdom Monera, and the unicellular eukaryotic organisms were placed in kingdom Protista. Kingdom Protista has brought together Chlamydomonas, Chlorella, which was earlier placed in algae within plants and both having cell walls, with Paramecium and Amoeba, which were earlier placed in animal kingdom which lacked cell wall. It has put together organisms which in earlier classifications were placed in different kingdoms. This happened because the criteria for classification changed. This kind of changes will take place in future too, depending on the improvement in our understanding of characteristics and evolutionary relationships. Over time, an attempt has been made to evolve a classification system which reflects not only morphological, physiological and reproductive similarities, but is also phylogenetic, that is, it is based on evolutionary relationships. In this chapter, we will study characteristics of kingdoms Monera, kingdom Protista, kingdom Fungi of Whittaker's system of classification. The kingdom Plantae and Animalia, commonly referred to as plant and animal kingdoms, respectively, will be dealt separately in chapters 3 and 4. So, first is kingdom Monera. Bacteria are the sole members of the kingdom Monera. They are the most abundant microorganisms. Bacteria occur almost everywhere. Hundreds of bacteria are present in a handful of soil. They also live in extreme habitats such as hot springs, deserts, snow and deep oceans, where very few other life forms can survive. Many of them live on other organisms as parasites. Bacteria are grouped under 4 categories based on their shape. The spherical shaped coccus, plural cocci. The rod shaped bacillus, plural bacilli. The comma shaped vibrium, plural vibrio, and the spiral shaped spirillum, plural spirilla. So you can see the diagrams: spherical shaped cocci, the rod shaped bacilli, the spiral shaped spirilla, and the comma shaped vibrio. These are the different types of bacteria on the basis of shapes. Though the bacterial structure is very simple, they are very complex in behavior. Compared to many other organisms, bacteria as a group show the most extensive metabolic diversity. Some of the bacteria are autotrophic, that is, they synthesize their own food from inorganic substrates. They may be photosynthetic autotrophic or chemosynthetic autotrophic. The vast majority of bacteria are heterotrophs, that is, they depend on other organisms or on dead organic matter for food. Archaebacteria. These bacteria are special since they live in most harsh habitats such as extreme salty areas that are called as halophiles, hot springs, thermoacidophiles, and marshy areas, methanogens. Archaebacteria differ from other bacteria in having a different cell wall structure, and this feature is responsible for this survival in extreme conditions. Methanogens are present in the gut of several ruminant animals such as cows and buffaloes, and they are responsible for production of methane biogas from the dung of these animals. eubacteria. These are thousands of different eubacteria or true bacteria. They are also called as true bacteria, and they are characterized by presence of rigid cell wall. If motile, and if they are motile, then they have a flagellum, a structure called as flagellum. The cyanobacteria, also referred to as blue-green algae, have chlorophyll similar to green plants and are photosynthetic autotrophs. The cyanobacteria are unicellular, colonial or filamentous. Freshwater marine or terrestrial algae. The colonies are generally surrounded by gelatinous sheath. They often form blooms in polluted water bodies. Some of these organisms can fix atmospheric nitrogen in specialized cells called heterocysts, example nostoc and annabina. Chemosynthetic autotrophic bacteria oxidize various inorganic substances such as nitrates, nitrites and ammonia and use the released energy for their ATP production. They play a great role in recycling nutrients like nitrogen, phosphorus, iron and sulfur. Heterotrophic bacteria are most abundant in nature. The majority are important decomposers. Many of them have a significant impact on human affairs. They are helpful in making curd from milk, production of antibiotics, fixing nitrogen in legumes, roots, etc. Some are pathogens causing damage to human beings, crops, farm animals and pets. Cholera, typhoid, tetanus, citrus canker are well-known diseases caused by different bacteria. So I know that cholera, typhoid and tetanus, these are caused to animals, these diseases, and citrus canker is mainly caused to the plants. Cholera is caused by the Vibrio cholerae, that is comma-shipped bacteria. Typhoid is Salmonella typhi. Tetanus is Clostridium tetani. And citrus canker is one of the plants. Okay, it is caused to one of the plants. Bacteria reproduce mainly by fission. Sometimes under unfavorable conditions, they produce spores. They also produce by sort of sexual reproduction by adopting a primitive type of DNA transfer from one bacterium to another. So you can see here the dividing bacteria where the DNA is getting divided, and first it undergoes fission. So first it is first the nucleus gets divided, that is karyokinesis, and then the first the nucleus gets divided, that is called as karyokinesis, and then the cytoplasm gets divided, that is called as cytokinesis. So this you can get that DNA is getting separated, and here you can see the figure of the filamentous blue-green algae, which is cyanobacteria, which is nostoc, and it has a sheath or covering called as mucilaginous sheath, and this structure in it is called as heterocyst. Okay. The mycoplasma are organisms that completely lack a cell wall. They are the smallest living cells known and can survive without oxygen. Many mycoplasma are pathogenic in animals and plants. Let us get to the kingdom Protista. All single-cell eukaryotes are placed under Protista, but the boundaries of this kingdom are not well defined. What may be a photosynthetic protestant to one biologist may be a plant to another. In this book we can include chrysophytes, dinoflagellates, euglenoids, slime molds and protozoans under Protista. Members of Protista are primarily aquatic. This kingdom forms a link with others dealing with plants, animals and fungi. Being eukaryotes, the protestant cell body contains a well-defined nucleus and other membranes-bound organelles. Some have flagella or cilia. Protists reproduce asexually and sexually by a process involving cell fusion and zygote formation. Chrysophytes. This group includes diatoms and golden algae, that is desmides. They are found in fresh water as well as in marine environments. They are microscopic and float passively in water currents, that is plankton. Most of them are photosynthetic. In diatoms, the cell walls form two thin overlapping shells which fit together as in a soap box. The walls are embedded with silica and thus the walls are indestructible. Thus, diatoms have left behind a large amount of cell wall deposits in their habitat. This accumulation over billions of years is referred to as diatomaceous earth. Being gritty, this soil is used in polishing, filtration of oils and syrups. Diatoms are the chief producers in the oceans. Okay. So, next is dinoflagellates. These organisms are mostly marine and photosynthetic. They appear yellow, green, brown, blue or red depending on the main pigments present in their cells. The cell wall has stiff cellulose plates on the outer surface. Most of them have two flagellae. One lies longitudinally and the other transversely in the furrow between wall plates. Okay. Very often read dinoflagellates, example goniolax. So example of dinoflagellates is goniolax. Undergo such rapid multiplication and they make the sea appear red tides. Okay, so this is due to red. Okay, so we know that these are the bacteria only and it is red dinoflagellates. So example is goniolax. Undergo such rapid multiplication that they make the sea appear red. That is called as red tides. Toxins released by such large members may even kill other marine animals such as fishes. So we can see here, we must read first theory and then I'll show you the pictures. Okay, the third is euglenoids. Majority of them are freshwater organisms found in stagnant water. Instead of a cell wall, they may have a protein-rich layer called pellicle, which makes their body flexible. They have two flagella, a short and a long one. Though they are photosynthetic in presence of sunlight, when deprived of sunlight, they behave like heterotrophs by predating on other small organisms. Interestingly, the pigments of euglenoids are identical to those present in higher plants, example euglena. Then fourth is slime molds. Slime molds are saprophytic protists. The body moves along decaying twigs and leaves engulfing organic material. Under suitable conditions, they form an aggregation called plasmodium, which may grow and spread over several feet. During unfavorable conditions, the plasmodium differentiates and forms fruiting bodies bearing spores at their tips. The spores possess true walls. They are extremely resistant and survive for many years. Even under adverse conditions, the spores are dispersed by air currents. Okay, so now let us see that we learned about the kingdom Protista. Okay, yes, first we learned about the kingdom Monera. Okay, then we learned about the kingdom Protista. Okay, in that we learned about different classes, and then we learned then are the protozoans, which are under the kingdom Protista only. So all protozoans are heterotrophs and live as predators or parasites. They are believed to be primitive relatives of animals. There are four major groups of protozoans. Amoeboid protozoans. These organisms live in fresh water, sea water or moist soil. They move and capture their prey by putting out pseudopodia, false feet, as in amoeba. Marine forms have silica shells on their surface. Some of them, such as entamoeba, are parasites. Entamoeba, or we can say entamoeba. It's not entamoeba, it is entamoeba or entamoeba. Okay. Flagellated protozoans. The members of this group are either free living or parasitic. They have flagella. The parasitic forms cause diseases such as sleeping sickness, that is trypanosoma. Okay, ciliated protozoans. These are aquatic, actively moving organisms because of the presence of thousands of cilia. They have a cavity or gullet that opens to the outside of the cell surface. The coordinated movement of rows of cilia causes the water laden with food to be stirred into the gullet. Example, Paramoecia. Sporozoans. This includes diverse organisms that have an infectious spore-like stage in their life cycle. Most notorious is Plasmodium, malarial parasite, which causes malaria, a disease which has staggering effect on human population. I was diagnosed with malaria when I was, I guess, 5 to 6 years. No, I was in first standard and I was diagnosed with malaria. So I was on the bed rest, and I was applied with the saline. Yeah, so it was like very terrifying moment in my life. I cannot forget it. So let us learn about the kingdom fungi. The kingdom fungi constitute a unique kingdom of heterotrophic organisms. They show a great diversity in morphology and habitat. You must have seen fungi on a moist bread and rotten fruits. The common mushroom you eat and toadstools are also fungi. White spots seen on mustard leaves are due to a parasitic fungus. Some unicellular fungi, example yeast, are used to make bread and beer. Other fungi cause diseases in plants and animals. Wheat rust causing Puccinia, Puccinia is an important example. Some are the source of antibiotics, example penicillium. Fungi are cosmopolitan and occur in air, water, soil and on animals and plants. They are preferred to grow in warm and humid places. Have you wondered why we keep food in the refrigerator? Yes, it is to prevent food from going bad due to the bacterial or fungal infections. With the exception of yeasts, which are unicellular, fungi are filamentous. Their bodies consist of long, slender threads-like structures called hyphae. The network of hyphae is known as mycelium. Some hyphae are continuous tubes filled with multinucleated cytoplasm. These are called let me repeat it. These are called coinocytic hyphae. Coinocytic hyphae. Again, I'll read this sentence. Some hyphae are continuous tubes filled with multinucleated cytoplasm. These are called coinocytic hyphae. Coinocytic hyphae. Others have septae or cross walls in their hyphae. The cell walls of fungi are composed of chitin and polysaccharides. Most fungi are heterotrophic and absorb soluble organic matter from dead substances and hence are called saprophytes. Those that depend on living plants and animals are called parasites. They can also live as symbionts in association with algae as lichens and with the roots of higher plants as mycorrhizae. Okay. Reproduction in fungi can take place by vegetative means, fragmentation, fission and budding. Asexual reproduction is by spores called conidia or sporangiospores or zoospores, and sexual reproduction is by oospores, ascospores and basidiospores. The various spores are produced in distinct structures called fruiting bodies. Okay, so let us repeat this paragraph. It has many new words that I don't know. Okay, so reproduction in fungi can take place by vegetative means, that is fragmentation, fission and budding. Asexual reproduction is by spores called conidia or sporangiospores or zoospores, and sexual reproduction is by oospores, ascospores and basidiospores. The various spores are produced in distinct structures called fruiting bodies. The sexual cycle involves the following three steps: fusion of protoplasms between two motile or non-motile gametes called plasmogamy, fusion of two nuclei called karyogamy, nuclei means nucleus, it's plural form of nucleus, meiosis in zygote resulting in haploid spores. So there are three steps. Stage 1 is of fusion of protoplasms between two motile or non-motile gametes called plasmogamy. Second stage is of fusion of two nuclei called karyogamy, and third stage is meiosis in zygote resulting in haploid spores. When a fungus reproduces sexually, two haploid hyphae of compatible mating types come together and fuse. In some fungi, the fusion of two haploid cells immediately results in diploid cells, that is 2N. However, in other fungi, ascomycetes and basidiomycetes, I will repeat again, in other fungi like ascomycetes and basidiomycetes, an intervening dikaryotic stage, n plus n, that is 2 nuclei per cell, occurs. Such condition is called a dikaryon, and the phase is called dikaryophase of fungus. Later the parental nuclei fuse and the cells become diploid. The fungi form fruiting bodies in which reproduction division occurs, leading to formation of haploid spores. The morphology of the mycelium mode of spore formation of fruiting bodies form the basis for the division of kingdom into various classes. Phycomycetes. The next unit is of phycomycetes. Members of phycomycetes are found in aquatic habitats and on decaying wood, in moist and damp places, or as obligate parasites on plants. The mycelium is aseptate and coenocytic. Aseptate, A-S-E-P-T-A-T-E, and coenocytic. Coenocytic, C-O-N-E-N-O-C-Y-T-I-C. Asexual reproduction takes place by zoospores, motile, which are motile, or by aplanospores, which are non-motile. These spores are endogenously produced in sporangium. A zygospore is formed by fusion of two gametes. These gametes are similar in morphology, that are called as isogamous, or dissimilar, that are called as anisogamous or oogamous. Some common examples of mucor or rhizopus, the bread mold mentioned earlier, and albugo, the parasitic fungi on mustard seed. Some common examples of phycomycetes are mucor, then rhizopus, the bread mold, that is called as rhizopus, and albugo. It is a parasitic fungi on mustard. Now ascomycetes, commonly known as sac fungi. The ascomycetes are mostly multicellular, penicillium, or rarely unicellular. It's like it is multicellular, which is in multicellular, the example is penicillium, or it is unicellular, example yeast, Saccharomyces. They are saprophytic decomposers, parasitic or coprophilous. They are, see, these ascomycetes are saprophytic, decomposers, parasitic or coprophilous, that is growing on dung. Dung means you know that it is poop. Mycelium is branched and septate. The asexual spores are conidia, produced exogenously on the special mycelium called conidiophores. Conidia on germination produce mycelium. Sexual spores are called ascospores, which are produced endogenously in sac-like asci, singular ascus. These asci, I guess it is asci, singular ascus. These asci are arranged in different types of fruiting bodies called ascocarps. Some examples are aspergillus, claviceps, and neurospora. Neurospora is used extensively in biochemical and genetic work. Many members like morels, that is M-O-R-E-L-S, and truffles, T-R-U-F-F-L-E-S, are edible and are considered delicacies. Basidiomycetes. So commonly known forms of basidiomycetes are mushroom, bracket fungi, or puffballs. They grow in soil, on logs and tree stumps, and in living plant bodies as parasites. Example, rusts and smuts. The mycelium is branched and septate. The asexual spores are generally not found, but vegetative reproduction by fragmentation is common. The sex organs are absent, but plasmogamy is brought by diffusion. I'll repeat it again. The sex organs are absent, but plasmogamy is brought about by fusion of two vegetative or somatic cells of different strains or genotypes. The resultant structure is dikaryotic, which ultimately gives rise to basidium. Karyogamy and meiosis take place in basidium producing 4 basidiospores. The basidiospores are exogenously produced on the basidium, plural basidia. The basidia are arranged in fruiting bodies called basidiocarps. Some common members are agaricus, that is mushroom, Ustilago, that is smut. I'll repeat again, Ustilago, that is smut. And Puccinia, let us say the spelling, Puccinia, Puccinia rust fungus. Okay, so then it is Deuteromycetes, commonly known as imperfect fungi because only the asexual or vegetative phases of these fungi are known. When the sexual forms of these fungi were discovered, they were moved into classes they rightly belong to. It is also possible that the asexual and vegetative stage have been given one name and placed under Deuteromycetes, and the sexual stage another and placed under another class. Later, when linkages were established, the fungi were correctly identified and moved out of Deuteromycetes. Once perfect sexual stages of members of Deuteromycetes were discovered, they were often moved to Ascomycetes and Basidiomycetes. The Deuteromycetes reproduce only by asexual spores known as conidia. The mycelium is septate and branched. Some members are saprophytes or parasites, while a large number of them are decomposers of litter and help in mineral cycling. Some examples are Alternaria coletotrichum and Trichoderma. See, I'll repeat the examples of Deuteromycetes are Alternaria, coletotrichum or coletotrichum and Trichoderma. And Trichoderma. Kingdom Plantae. Kingdom Plantae includes all eukaryotic chlorophyll containing organisms commonly called plants. A few members are partially heterotrophic, such as insectivorous plants or parasites. Bladderwort and Venus flytrap are examples of insectivorous plants, and Cuscuta is a parasite. The plant cells have an eukaryotic structure with prominent chloroplasts and cell wall mainly made of cellulose. You will study the eukaryotic cell structure in detail in chapter 8. Plantae includes algae, bryophytes, pteridophytes, gymnosperms and angiosperms. Life cycle of plants has two distinct phases: the diploid sporophytic and haploid gametophytic. Again, I'll repeat, life cycle of plants has two distinct phases: the diploid sporophytic and the haploid gametophytic, that alternate with each other. The lengths of haploid and diploid phases and whether these phases are free living or dependent on others vary among different groups in plants. This phenomenon is called alteration of generation. You will study further details of this kingdom in chapter 3. Kingdom Animalia. This kingdom is characterized by heterotrophic eukaryotic organisms that are multicellular and their cells lack cell walls. They are directly or indirectly depend on plants for food. They digest their food in an internal cavity and store food reserves as glycogen or fat. Their mode of nutrition is holozoic. By ingestion of food, they follow a definite growth pattern and grow into adults that have definite shape and size. Higher forms show elaborate sensory and neuromotor mechanism. Most of them are capable of locomotion. The sexual reproduction is by copulation of male and female, followed by embryological development. Salient features of various phyla are described in chapter 4. The last section is viruses, viroids, prions and lichens. In the 5 kingdom classification of Whittaker, there is no mention of lichens and some acellular organisms like viruses, viroids and prions. These are briefly introduced here. All of us have suffered the ill effects of common cold or flu. Know what effects viruses can have on us. Even if we do not associate it with our condition, viruses did not find a place in classification since they are not considered truly living. If we understand living as those organisms that have cell structure, the viruses are non-cellular organisms. They are categorized by having an inert crystalline structure outside the living cell. Once they infect a cell, they take over the machinery of the host cell to replicate themselves, killing the host. Would you call viruses living or non-living? Virus means venom or poisonous fluid. Dmitry Ivanovsky in 1892 recognized certain microbes as casual organisms of the mosaic disease of tobacco. These were found to be smaller than bacteria because they passed through bacteria-proof filters. M.W. Bijanare Bijarinek, sorry, I'll repeat it again, it first was wrong. M.W. Bijarinek in 1898 demonstrated that the extract of the infected plants of tobacco could cause infection in healthy plants and named the new pathogen virus and called the fluid as Contagium vivum fluidum, infectious living fluid. Again I'll repeat, M.W. Bijarinek in 1898 demonstrated that the extract of infected plants of tobacco could cause infection in healthy plants and named the new pathogen virus and called the fluid as Contagium vivum fluidum, which is infectious living fluid. W.M. Stanley in 1935 showed that viruses could be crystallized, and crystals consist largely of proteins. They are inert outside their specific host cell. Viruses are obligate parasites. In addition to proteins, viruses also contain genetic material that could be either RNA or DNA. No virus contains both RNA and DNA. A virus is a nucleoprotein and the genetic material is infectious. In general, viruses that infect plants have single-stranded RNA and viruses that infect animals have either single or double-stranded RNA and double-stranded DNA. Okay, एक minute, just let me repeat. In general, viruses that infect plants have single-stranded RNA and the viruses that infect animals have either single or double-stranded RNA or double-stranded DNA. Bacterial viruses or bacteriophages. Viruses that infect the bacteria are usually double-stranded RNA, DNA viruses. I'll repeat it again. It's something complex, very complex, but it will be simple when I'll understand it. Bacterial viruses or bacteriophages, viruses that infect the bacteria, are usually double-stranded DNA viruses. The protein coat called capsid, made of small subunits called capsomeres, protects the nucleic acid. These capsomeres are arranged in helical or polyhedral geometric forms. Viruses cause diseases like mumps, smallpox, herpes, and influenza. AIDS in humans, AIDS in humans is also caused by a virus. In plants, the symptoms can be mosaic formation, leaf rolling and curling, yellowing and vein clearing, dwarfing and stunted growth. Okay, I remembered. I was just remembering the full form of AIDS, acquired immunodeficiency syndrome. So now see, viroids. In 1971, T.O. Diner discovered a new infectious agent that was smaller than viruses and caused potato spindle tuber disease. It was found to be a free RNA. It lacked the protein coat that is found in viruses, hence the name viroid. The RNA of the viroid was of low molecular weight. Prions. I'll repeat it. In modern medicine, certain infectious neurological diseases were found to be transmitted by an agent consisting of abnormally folded protein. The agent was similar in size to viruses. These agents were called prions. The most notable diseases caused by prions are bovine spongiform encephalopathy, BSE. I'll repeat it again, bovine spongiform encephalopathy, BSE, commonly called mad cow disease in cattle, and its analogous variant CR-Jacob disease, that is CJD, in humans. Lichens. Lichens are symbiotic associations, that is mutually useful associations between algae and fungi. The algal component is known as phycobiont. I'll repeat it again. The algal component of lichen is known as phycobiont, and fungal component as mycobiont, which are autotrophic and heterotrophic respectively. Algae prepare food for fungi, and fungi provide shelter and absorb mineral nutrients and water for its partner. So close is their association that if one saw a lichen in nature, one would never imagine that they had two different organisms within them. Lichens are very good pollution indicators. They do not grow in polluted areas. Okay, what are lichens? They are very good pollution indicators. They do not grow in polluted areas. Okay, by summary of this lesson, biological classification of plants and animals was first proposed by Aristotle on the basis of simple morphological characters. Linnaeus later classified all living organisms into two kingdoms, Plantae and Animalia. Let me highlight it. Aristotle on the basis of simple morphological characters. Then was Linnaeus, later classified all living organisms in Plantae and Animalia in two kingdoms, Plantae and Animalia. Whitaker proposed an elaborate five-kingdom classification, Monera, Protista, Fungi, Plantae and Animalia. The main criteria of five-kingdom classification were cell structure, body organization, mode of nutrition and reproduction, and phylogenetic relationships. In the five-kingdom classification, bacteria are included in kingdom Monera. Bacteria are cosmopolitan in distribution. These organisms show the most extensive metabolic diversity. Bacteria may be autotrophic or heterotrophic in their mode of nutrition. Kingdom Protista includes all single-celled eukaryotes such as chrysophytes, dinoflagellates, euglenoids, slime molds, and protozoans. Protists have defined nucleus and other membrane-bound organelles. They reproduce both asexually and sexually. Members of kingdom Fungi show a great diversity in structures and habitat. Most fungi are saprophytic in their mode of nutrition. They show asexual and sexual reproduction. Phycomycetes, ascomycetes, basidiomycetes, deuteromycetes are the four classes under this kingdom. The Plantae includes all eukaryotic chlorophyll containing organisms, algae, bryophytes, pteridophytes, gymnosperms and angiosperms are included in this group. The life cycle of plants exhibit alternation of generations, gametophytic and sporophytic generations. The heterotrophic eukaryotic multicellular organisms lacking a cell wall are included in the kingdom Animalia. The mode of nutrition of these organisms is holozoic. They reproduce mostly by sexual mode. Some acellular organisms like viruses and viroids as well as lichens are not included in five-kingdom classification of the five-kingdom system of classification. Okay, I remember that we were left to see the diagram. So from first page we'll try to examine all the diagrams that are given in this textbook. Okay, so let us see. Okay, so we had seen this one. Okay, four types. The bacteria are classified into four types on the basis of shapes. So they are cocci, spherical shaped cocci, rod shaped bacilli, spiral shaped spirilla, and comma shaped vibrio. The next diagram is of the filamentous blue-green algae called as nostoc. We have discussed about this earlier. Then the dividing bacteria, it is either by fission or sometimes producing spores. Then we are okay. So here we have not arrived at this figures. Dinoflagellates is this is the diagram of the dinoflagellates. Okay, dinoflagellates. Okay, then this is the diagram of the euglena. And this has, see, dinoflagellates also has I guess it has two flagella, and euglena also has two flagella. The C1 is slime mold. See, this diagram is of the slime mold. This is euglena and this is dinoflagellate. Okay, and this one is paramecium. Okay, okay, it's like the slipper that we wear, paramecium. Okay, it has cilia, ciliated structure. Okay, yes. Then there is figure of I guess it is fungi. First is mucor. Fungi named as mucor. Here we can see the dot-like structures which are called as sporangia, and this is called as hyphae. The second figure is of aspergillus. Okay, so this is aspergillus, and here we can see the dot-like structures here. See, here you can see. Let us take the red pen so that it will be visible to you, and I'll take some more. And just make it thicker. See this dot-like structures which are present here. See, you can see here. This is a sporangia which is shown, and this sporangia structure produces spores, and it is a reproductive part of this fungi. And the third one is agaricus. Okay. Then the figure is of. Okay, let us see the figure of the tobacco mosaic virus, that is TMV. This first figure, this one. This first figure is of tobacco mosaic virus, and it has RNA and capsid. Okay, and the second figure is of bacteriophage, which has a head, which has a collar, this one, and also has a sheath. Okay, so I guess we have completed with all the diagrams. Let us read all the questions. Exercises. First question, discuss how classification systems have undergone several changes over a period of time. State two economically important uses of heterotrophic bacteria, archaebacteria. What is nature of cell walls in diatoms? Fourth question, find out what do the terms algal bloom and red tides signify. Fifth question, how are viroids different from viruses? Sixth is, describe briefly the four major groups of protozoa. Seventh is, plants are autotrophic. Can you think of some plants that are partially heterotrophic? Eighth is, what do the terms phycobiont and mycobiont signify? Ninth is, give a comparative account of the classes of kingdom fungi under the following. First is mode of nutrition and second is mode of reproduction. 10th question is, what are the characteristic features of euglenoids? 11th is, give a brief account of viruses with respect to their structure and nature of genetic material. Also name 4 common viral diseases. And 12th question is, organize a discussion in your class on topic, are viruses living or non-living? And finally, we have come to an end of this lesson, the biological classification, and I will definitely read all the 22 chapters of class 11, and it will be very beneficial for me, as we know, finally, as we know at last, the concluded part, that the NCERT is our god for the NEET aspirants. So I will do it, I will become doctor, and I'll show the world what is sensory level. Thank you.